Okay, well, I think we'll make a start. Welcome, everybody. This is the Health Research from Home webinar series. It's our fifth webinar. And my name's Will Dixon. I'm the lead investigator for Health Research from Home. I'm a professor of digital epidemiology, and I'm a consultant rheumatologist in Salford. Together with Coral, we're hosting the webinar today. We have a great program. Going to do some housekeeping first of all. Then we got two talks. First from Val Hamy talking about discovering digital biomarkers to support drug development in rheumatoid arthritis. And then Aidan Acquah, PhD student at Oxford, talking about the use of wrist-worn accelerometry to predict Parkinson's disease. We're going to take question and answers at the end of those two talks. Afterwards, we'll move to an update about one of our two driver projects. These are projects that are looking to link patient-generated health data collected via smartphones and wearables typically together with other NHS data sources. And we're following two projects through the course of the 3 years. And then we'll end with some brief announcements. But first of all, the housekeeping. The webinars are being recorded. We will make those recordings available on the Health Research from Home YouTube channel at the end, so you can access them and rewatch them there. And that will be trimmed so the Q&A session isn't part of the recording. During the course of the presentations, please do make use of the question and answer button in the webinar. So you can submit your questions through the course of the talk, and then we'll take questions both from the Q&A box and from hands raised. You'll be muted throughout the talk, the whole of the audience will, but you can raise your hand and we can unmute people during the Q&A. So you can submit your questions or you can raise your hand. And then at the end of the webinar, we're going to take some feedback from you. Usually we've done that about the webinar program, but we're going to ask you a couple of questions about our forthcoming grant writing retreat. So that's it from me. That's the end of the housekeeping. And I'd like to invite our first speaker, Val Hamy, to deliver his talk. So thanks very much, Val. Over to you.
Thank you very much, Will. So just going ahead and sharing my screen. Okay. All right. Well, great to be here. Thank you very much for the opportunity to present this work. I will start with a bit of context around the use of digital biomarkers in drug development. So first of all, what are digital biomarkers? These are measurements of physiology and behavior that we capture using digital technologies such as sensor, mobile phones, mobile apps, or wearable devices. Our team at GSK operates within a large organizational matrix, engaging with a variety of stakeholders across multiple disciplines. We do collaborate closely with digital health, real-world evidence, and clinical operations, focusing on technology, data, and operationalization aspects. We also work with research, clinical sciences, and patient-centered outcomes regarding the added value to clinical development programs, ensuring that patient outcomes are the forefront of our work. Then also working with biostatistics and regulatory affairs around quantitative evidence and validation issues, ensuring that our methodologies meet regulatory standards. This multidisciplinary interaction allows us to leverage expertise from various fields to potentially transform how we develop and launch novel medicine. Now, one challenge we face somehow with the digital biomarkers is that there are very many potential applications. And one can think about monitoring or diagnostic or prognostic and many others. Hence, there is a need to focus on a few areas that are the most impactful. And for this, we shortlisted a set of key opportunities aligned with our medicine development pipeline where we can add value. The first one is around treatment efficacy assessment. So in this context, digital biomarkers can add value in two different ways, actually. First, we can develop digital endpoints which have higher sensitivity to change over time compared to traditional clinical outcomes, and thus allowing for more accurate monitoring of symptoms, which in turn can support decision-making. Secondly, we can develop patient-centric digital endpoints that are able to objectively quantify the value of a new medicine. For example, around aspects that are known to be important to patients, such as fatigue, pain, or mobility, which are typically captured using patient-reported outcomes that provide valuable information on how patients feel but it's possible to complement that information with an additional layer of objective data that describes or highlights how patients function. It is precisely this idea that we will be focusing on later in the presentation in the context of rheumatoid arthritis. Other opportunities exist in the safety space. First, in the context of decentralized clinical trials, where key safety measures can be taken directly from patients, from the patient's home, using digital technology, so that there is no longer the need to be at a clinical site. And of course, if patients have access to that technology at home, it might be possible to monitor them continuously, or at least increase the frequency of the measurements for additional safety-related insights. Then we can think about patient stratification, where digital biomarkers can provide input to disease progression models, allowing for the characterization of subgroups of patients that might benefit the most from a new treatment. And the last opportunity is around companion devices, which corresponds to the use of digital technology post-medicine approval. So we can collect real-world data from patients who have been prescribed the medicine and develop potentially or provide information that may be relevant to the physician to inform treatment decision. Then another important question relates to the views of regulators on digital health technology. And the FDA released a set of guidance on how to best incorporate this type of technology. Also, the Digital Medicine Society, DIME, developed a framework for digital endpoint validation, which is summarized in this slide, and aligns with the FDA guidance. So the first stage is concept elicitation, which involves understanding what matters the most to patients, whether or not this concept of interest can be captured with a digital health technology, and if so, how. Then once a DHT or digital health technology has been selected, we need to make sure of the technical feasibility and the usability from a patient perspective. Are the patients able to and willing to use the technology? Is it operationally feasible to implement this technology in a study, for example, in terms of data transmission or data quality? And then assuming these points are fully addressed, the next stage is the analytical validation, which evaluates the performance of an algorithms that transform raw data into measures reflecting the identified concept of interest. This includes checking the accuracy and reliability of the measures. Measures that are analytically valid then need to be assessed for clinical validity. First, in terms of construct-related validity, that would be, does the measure correlate with other patients' reported or clinical outcomes of the same concept? And then in terms of definition of a minimum change that can be interpreted as meaningful to patients. In other words, what is the magnitude of change that need to be observed in that specific measure to be able to say that the medicine is beneficial for patients? And finally, once we have all these aspects covered, we should have a digital endpoint that is pretty much ready for deployment. So now let's move on to our specific use case, starting with a recap on the disease. Rheumatoid arthritis, or RA, is a chronic autoimmune disease that is characterized by persistent inflammation of joints with chronic pain, stiffness, and swelling, which leads to deformities and inability to perform daily tasks or daily activities. Another characteristic of RA is that it's heterogeneous, with daily symptoms fluctuation, for example, with more severe stiffness typically experienced during the first couple of hours after getting up in the morning compared with the rest of the day, and also fluctuations taking place over the course of several days. This takes us to some of the current challenges with RA, with a lack of fit-for-purpose measure that can accurately capture symptom relief or improvements in physical function. This challenge may be tackled using digital health technologies that enable continuous monitoring of symptoms that matter to patients, including morning stiffness, mobility, fatigue, and sleep quality. Another challenge relates to the need for sensitive endpoints that can objectively demonstrate treatment efficacy. And here the wealth of information provided by digital health technology can be unlocked through advanced analytics and address this particular challenge. And in this context, the watch and mobile devices for the assessment of rheumatoid arthritis burden on daily life compared to patient-reported outcomes, or WeaRAble-Pro for short, was an observational study where digital data collected using an Apple Watch and integrated bespoke app on an iPhone was used to characterize the impact of RA on daily life. The study was run in partnership with Analysis Group and with contribution from partner groups at the University of Oxford. It included 60 participants with 30 patients with moderate to severe RA and 30 matched healthy controls with matching performed on age, gender, and ethnicity. Following an initial visit at the clinic where all the participants were given the devices and were trained on how to use the app, data was collected for a total duration of 14 days. During this period, several types of data were collected via the app, including PROs capturing information on sleep quality, on the overall impact of the disease, on quality of life, as well as pain and stiffness. And a small number of predefined activities were also called guided tests, were administered to evaluate the impact of the disease on specific function or type of motion. These were performed twice a day, once in the morning and once in the afternoon. Data on physical activity was also collected continuously using the accelerometer in the Apple Watch, and the use of research kits allowed us to access the raw sensor data and not only the pre-processed summaries that are typically provided by Apple. All that information was then processed and analyzed with the aim to characterize the impact of RA on daily life. So this slide shows a few examples of PRO instruments that were administered through the app, including questionnaires, visual analog scales, and also a joint pain map, which is presented in this video on the right. So this is a useful tool to provide an accurate view of the level of pain that participants were experiencing at each joint. By navigating through that interactive dynamic body map on the screen, they could highlight the painful joints and provide some detailed information on the level of pain. And all the corresponding data provided reference material in the assessment of digital sensor-derived measures. So now let's take a look at the guided tests. WeaRAble-Pro included 5 guided tests, targeting wrist range of motion, hand dexterity, gait, and specific transitions from sitting to standing and from sitting to lying. Next slide focuses on the lie to stand transition test. Why is this an important concept? Well, this type of motion matters to patients as it is part of everyone's daily activities. For example, when getting up from the bed in the morning, and it can be heavily impacted by the disease. This type of motion mobilizes different joints too, and as such, it can be related to overall mobility, which is an important aspect of quality of life. Here we see the on-screen guidance provided in the app. Participants were instructed to perform the lie to stand transition twice with the phone strapped into their right thigh. So now if we look at this practical example with video that can be seen at the top left of the screen, we see someone performing the exercise. And using the data collected from the iPhone sensors, it is possible to recreate the device 3D motion to see the change in orientation over time. This is useful for visualization purpose and potentially quality checking. But the information that we are really interested in is the representation of the acceleration signal through time along the 3 directions of space. This is what contains the information that we want to access regarding the transition times from lying to standing and from standing to lying, as we can see here. So I should mention that although this looks pretty simple in this example, detecting such transitions requires expert knowledge and the development of bespoke signal processing algorithms. But then looking at an aggregation of these measures across the entire study population and across the 14 days of data collection, we can start looking into their analytical characteristics. Considering the distribution of the the mean lie to stand transition time in both participant groups, we can see that RA patients are generally slower with more variability compared with healthy controls. And this variability is not unexpected because of symptoms fluctuations and the potential differences between morning and afternoon. This may also reflect the heterogeneity of symptoms across patients. Now, looking into construct validity of this measure, we compare the values with PRO data on fatigue and morning stiffness, and we can see that the mean transition time seems to be longer with increasing level of fatigue and stiffness. Another interesting observation is the fact that for both PROs, healthy controls reported some level of either fatigue or stiffness, but this doesn't seem to be impacting the transition time this much. All these findings were confirmed through statistical analysis. Obviously, the small number of participants limits the generalizability of our observations, but there may be a signal here that we're looking in the right direction. Now, moving on to the watch data analysis. So in this case, we are considering accelerometer data recordings over 14 days with the objective to characterize daily patterns of activity. We looked at 4 domains of activity, of physical activity, which are total volume of activity, activity fragmentation, morning activity, and nighttime restlessness. This particular analysis was performed as part of a research project in collaboration with the Oxford wearables group and the computational health informatics lab at the University of Oxford. As opposed to the Light-to-Stand test data that I showed earlier, with a very specific action performed over a short period of time, we're now looking at several days of any activity. Hence, the approach to analyzing this data was quite different. In this case, a deep convolutional neural network model was used to perform human activity recognition. And the examples in the slides represent data from a healthy control on the left and an RA patient on the right. Each row corresponds to a specific day with black solid line showing the acceleration magnitude and colored areas for each type of activity detected, with blue for sleep, red for sedentary, yellow for light activity, and green for moderate to vigorous physical activity, or MVPA. We also have vertical dotted lines indicating participants' self-reported sleep times, and blank areas corresponding to device charging time. Upon visual inspection, we can see that the RA patient is a lot more sedentary than the control, with very limited MVPA. And by aggregating this high-level data on detected activity, it is possible to derive profiles for each group with information on the probability of a specific activity type to take place depending on the time of the day. And if we overlay the profiles for both groups, then a few differences can be observed, especially in terms of sleep and MVPA. This is very qualitative at this point, but what is really interesting is that this information can guide us into the derivation of more specific measures, and that's exactly what we did next. Here are a few examples where the daily average time spent in MVPA is shown on the left and appears to really differentiate between healthy controls and the 2 RA subgroups, moderate RA and severe RA. The measure in the middle, showing reflecting morning activity, seems to be better at differentiating between controls and moderate RA. And the measure shown on the right, which is related to prolonged sedentary periods, captures differences between the 2 RA subgroups. So there is some signs of some kind of complementarity between the measures. And as a next step, what we did was that we explored how multivariate analysis could combine this information from different sources. First, we looked into how to combine measures derived from the watch data and the guided tests as input to a binary classification model, separating controls and RA patients. So here, the actual performance of the model is not really what matters, because in clinical studies, we would know who has RA and who doesn't. However, this is what we can learn from the data that is useful. Looking at the performance metrics, it's actually the combination of the 2 data sources that provides the best classification. Then, when we look at the features importance, we do see that the mean transition time from standing to lying is one of the key measures driving class assignment. Then we repeated the same exercise, but adding PRO scores for the classification of moderate versus severe RA. There's no point including control groups in this case, because we know that PROs are already really good at differentiating between healthy and RA. But in this context, we wanted to highlight the fact that adding information from the digital source could actually boost the sensitivity to severity compared to PROs alone. And that's what is shown in the performance model, in the model performance plot. Interestingly, the feature importance table shows a PRO score at the top as the top feature, with hand dexterity, wrist motion, and walk-related measures coming just next. Going further, we explored how the combination of PRO scores and digital measures could be used to estimate a separate disease severity score named the RAPID-3, which was administered during the baseline visit at the clinic. And we found that the model is doing a pretty good job, working pretty well, except for a few outliers. In fact, when we tried to get some more context around these estimates, using data from the joint pain map that I mentioned earlier and looking at the cumulative joint pain across days, the findings were interesting. In this first example in the top right, where the RAPID-3 score was accurately estimated, a fairly high and stable level of pain had been reported. But now if we look at other examples, especially the one at the bottom where the model importantly underestimated the RAPID-3 score, then looking at the joint pain map, we see that there's hardly any pain reported after baseline, which may be a sign that actually the model is pretty robust. Again, this is very exploratory at this stage, but these observations seem to be showing some sign that we're heading towards the right direction in terms of building some kind of composite endpoints by combining different features, different measures. Very briefly, we could find, well, actually, an extension of this work of the WeaRAble-Pro Study data analysis consisted of exploring the same measures as those derived from the Apple Watch data in a large-scale data set such as the UK Biobank, which Aidan will talk about later in this presentation. I won't spend too much time on this in the interest of time, but basically what we could find, we could find about 1,000 participants who had been diagnosed with RA and for whom physical activity monitoring data was available. And these were matched in the same way as in the WeaRAble-Pro study with non-RA controls. And using this similar representation, qualitative representation of daily activity profiles, we could see, again, similar differences between the two groups. We also performed some quantitative analysis of specific measures, but in the interest of time, I won't describe those today. So in summary, what did we learn? We found that a number of digital measures of RA show promising performance from a construct-related validity point of view. And that's including an ability to differentiate between controls and RA groups, as well as convergent validity with PRO scores. An extension of the study looking at a large-scale data set led to similar findings. We also touched on multivariate analysis combining measures from different sources to provide a more accurate representation of the disease. And this may be the first step towards the creation of a composite endpoint for RA. Thinking about the validation framework, one missing piece really is the assessment of sensitivity to change, which would require the access to longitudinal data and should be the focus of a future study. I didn't talk about the feasibility aspects either, but we had excellent patient compliance. Of course, this was a very small study of only 14 days in a small group of patients, but we saw promising results from the point of view of compliance and adherence. And that concludes my presentation. Thank you very much.
Val, excuse me. Thank you so much, Val. That was a fascinating talk. As a rheumatologist, it's particularly interesting as I see patients with rheumatoid arthritis every week, and yet we only see them once every 6 months and try and understand how their disease has been by asking the question, how have you been in the last 6 months? Just a reminder to the audience that you can submit questions via the Q&A chat box. But we're going to move straight on to the second talk. In Health Research Room Home, we like to allow everyone onto the stage, and we specifically have the second talk from somebody who's an earlier career researcher. So it's a real pleasure to introduce Aidan, PhD student at Oxford. So Aidan, over to you. We can see your slides fine, Aidan. It looks like you're on mute currently.
Yes, I was just trying to find the unmute button. Thank you. Okay. Thank you so much. Good afternoon, all. And today I'll be presenting my work regarding the use of wrist-worn accelerometry to predict Parkinson's disease. And this is the collaboration of many departments within Oxford and also supported by GSK. So, good. first slide. So Parkinson's disease is the second most common and fastest growing neurodegenerative disease worldwide, with 10 million cases, and numbers are continuing to grow. There is no known cure. However, there are various treatments that often limit the symptoms, but are not perfect. And so this has encouraged researchers to look into early detection of the disease as a new source of research. So illustrated across, is some various symptoms of Parkinson's disease. However, these can be both motor and non-motor related. But as highlighted, researchers are very much interested in early stages of the disease, particularly the prodromal phase, prior to a diagnosis, a clinical diagnosis. There are various slight motor signs that can give an indication of a pathway towards a disease that perhaps could be picked up through the use of wearables and digital biomarkers. So wearables offer a new dimension to health information. So this could be accelerometers or gyroscopes, PPG, that can monitor things like activity levels or heart rates. And this can be collected over long periods of time, collected at home during free living, and also can be a low burden to use in terms of research. And so this allows the potential for screening of the population, for monitoring of disease progression, and for reporting of drug efficacy. And so the motivation behind this work is that we would like to better identify at-risk populations of developing Parkinson's disease. And so perhaps we could do recruitment into monitoring studies of the prodromal PD population. And then we would also like to be able to better understand the risk markers for early Parkinson's disease. So what actual features of prodromal Parkinson's disease can actually be picked up and are true markers of the disease. However, the current gaps in the literature are that we're not sure which of these accelerometer-derived features are actual markers for Parkinson's disease, and which should we best target for a strong association to the disease, but also good communication to the general population. So as Val mentioned, we're looking at the UK Biobank, which is a cohort study of over 500,000 individuals, from which we have a variety of different information, which includes questionnaire data, genetic data, and wearables data. From this data, we are able to extract various risk factors, which includes things like age, sex, coffee intake, alcohol intake from the questionnaire data. We can develop polygenic risk scores that are extracted from the genetic data collected from the participants. And then also we can derive accelerometer-derived features from the wearables data. And the wearables collected was using an AX3 activity device, so a research-grade device for which we have the raw acceleration data in 3 axes over the course of 7 days. So from these risk factors, we can develop risk models, which use a combination of these risk factors. So for example, we have a demographic model here that could use a combination of the questionnaire-derived risk factors. However, we could also develop a risk model that uses factors like a PD-like gait score, which would produce a different survival probability from that risk model. So just exploring the accelerometer-derived risk factors a bit better. First, we have overall activity, which is a measure of the average acceleration of the Euclidean norm -1, a technical way of saying what's the overall average activity that is seen in the signal. However, we can use more complex features extracted using machine learning, as shown by Val once again. And this, for example, could be deriving the activity intensity composition derived using machine learning. And so this is where we have identified for this individual over the course of 7 days when the individual was asleep or sedentary, light, moderate, vigorous activity. And we can derive this into a composition score of the overall proportion of time spent in each activity. Finally, we can also do a PD-like gait score, in which we can compare the active walking windows of this individual to that of known diagnosed PD individuals. And this could give a greater indication of how similar their walking is to those with Parkinson's disease. So looking at the study population, the UK Biobank starts at with over 500,000 individuals. However, for this study, we are able to use 96,000 individuals, just over 96,000 individuals data that required that they did participate in the activity monitoring study. So we did have accelerometer data from them, that they had usable accelerometer data, that they didn't withdraw from the study, and they did not already have Parkinson's disease. This population on average was around 62 years old, was 56% female, and typically did drink more than once a week, and had never smoked. To extract the outcomes to determine whether these individuals did or did not develop Parkinson's disease, we use hospital inpatient and death data. And so from these 96,000 individuals, we have just over 400 incident PD cases found, with a median follow-up time of 7.9 years. So now looking at the results of this study. So first thing, we evaluated the performance of our risk models using concordance index, which is a measure of how well we were able to discriminate high risk and low risk individuals. So a high concordance index corresponds to a good performing model, and you expect it to cap at 1. So in this case, we see that using just demographic factors, so just things like age, sex, alcohol, coffee, and so on, we see a concordance index of 0.778. However, when creating a model which included also accelerometer-derived risk factors, the concordance index was much higher at 0.902. And this was found to be significant. So very good. So the conclusion, to put this into a practical application, let's say that we wish to conduct a sub-study from the UK Biobank Physical Activity Monitoring Study, in which we wanted to further investigate the participants of highest risk, to look deeper into those individuals, with the hope of getting as many individuals that are in the prodromal phase of PD as possible. Well, how many of those with the highest N risk would actually go on to have a PD diagnosis according to the different models? Well, if we take an example of just 300 individuals, so if we took the top 300 highest risk individuals, if we were to use a random model, we would only get 2 individuals that actually went on, we would expect to go on to have Parkinson's disease. And this is because it's quite a rare condition. However, using our model, using all risk factors, we would have 98 individuals of the 300 that actually went on to be in the prodromal phase of Parkinson's disease. And so we have greatly increased our chance of being able to identify those with that went on to go and have Parkinson's disease. So this research has shown provisionally that within the UK Biobank, the risk models that included accelerometer-derived risk factors had a higher C index in discriminating in the incident Parkinson's disease. And higher risk participants were better identified using these models. However, there are limitations to this study. We do not have external validation data sets at the moment. And so we're looking at data sets like the NHANES that might give the potential of being able to explore other high population and also have accelerometer data. We have limited case numbers and follow-up time. And it's also coupled with the fact that we've not used primary care data, which may also reveal some greater numbers of Parkinson's disease. There's great research by Dr. Leigh Clifton surrounding this and the potential of being able to add to the numbers of Parkinson's disease using primary care data. And so this is something we would look to do in the future. And other future work includes long-term monitoring of accelerometer-derived features, and then also sleep-related accelerometer-derived risk factors. But for now, thank you for listening, and thank you to all of my co-authors.
So thank you very much indeed, Aidan. 2 wonderful talks. But I do need to move on now to the third talk. As I said at the beginning, in Health Research from Home, we're running 2 driver projects that are trying to learn about how we join together patient-generated health data from smartphones and wearables together with other data sources. Nathan is involved in the second of these two driver projects, and we're going to hear updates throughout the 3 years as to how those two projects are getting on. So Nathan, over to you so we can hear where you're up to. Thank you.
Thanks, Will. I'll just share my screen. Okay. Yes, I'm going to be giving an update on the second driver project, which we introduced first in the September webinar. And this is looking at the consequences of long COVID on people's health within the participants of the COVID symptom study biobank. So as a recap of why we're doing this project, the background is that we know within the UK there's millions of people. I think the last estimate was around 2 million people who were still living with long-term symptoms after having COVID-19. However, most of these people have not been coded as having long COVID in their medical records. And we know actually that within that the healthcare record, there's biases in terms of who's been recorded so far that need to be addressed. So what we want to know, firstly, is how do our participant reported data match with what's in people's healthcare records? And we have two different forms of participant reported data. We have a prospective data from a smartphone app where people logged symptoms every day during the pandemic, and then retrospective survey questionnaire data. And we want to understand how these different data sources correlate with each other, kind of learn for future remote research. And then we want to look at, in the longer term, how people's symptoms in the early stages go on to affect their health in the longer term, and we can track that or monitor that using the healthcare record data. So our participants were recruited through the COVID symptom study smartphone app, which was an app started right at the start of the COVID-19 pandemic, where people could log lots of different symptoms on a daily basis. And what that allows us to do is look on an individual and then at a population level as to the patterns of symptoms that people had after having COVID-19. And on screen is an example of an individual showing the kind of different symptoms, how they come and go over time, and as well as whether people have symptoms or not on a given day. So we think there's lots to learn from this type of data for use in future remote research. And we want to know, like, how the way in which people report on an app, how does that then go on relate to how they use and interact with healthcare, and then longer term their outcomes in terms of their health. So that's the background. I'm now going to talk about like where we're up to at the moment, and this is going to be focusing on the process of linking our cohort data with NHS healthcare record data. We're doing this via a platform that started during the COVID-19 pandemic called the UK Longitudinal Linkage Collaboration, or I'll just refer to it as the LLC. And we're going through this onboarding process at the moment, and these are the kind of different steps, different stages of onboarding before we can start to join together the data sets. And just to kind of give people an insight into what's happening behind the scenes, the different milestones that we're going through. One of the first steps is for the LLC to review the consent and participant information materials that we have to date, to see if they're compatible or with how the LLC is set up, and review them and update them. The next steps for us are then to organise the opportunity for participants to opt out if they no longer want to link their data with healthcare data. We're starting the contracts process between the organisations. So the cohort is based at King's College London, LLC is based in University of Bristol, and there needs to be data transfer agreements in place, and that goes through the legal teams. And there's going to be help from the LLC with communications with our participants. As well as that, the LLC needs to go through a process of updating their ethics agreements based on how our cohort aligns with what they've agreed already, and if there needs to be any amendments to be made. The LLC also need to talk to NHS England, who are providing the healthcare record data, again, to see if there's any amendments or changes that they need to make in their agreement there. And it's then towards the end of the process that we as a cohort will be invoiced, and then it's only then after all these steps that we send our data to be uploaded and ingested onto the platform, where it's then linked with all the healthcare record data that the platform contains. So that said, we're still at this kind of early stage in the process, and I wanted to give kind of a more detailed idea of the timelines involved in case other people are thinking about this kind of process as well. So the timeline so far, we had our first informal meeting with the LLC to talk about we wanted to join, we were interested in kind of get the pitch from them. That was back in January of this year. But our first more formal meeting about the onboarding process was in the start of summer. We then shared materials as requested for review, and had another check-in towards the end of summer. But this material that we shared has only just been reviewed by a panel at the LLC in the last few weeks, and we're now waiting for the feedback, the minutes of the meeting, and things like that, so we can understand if any changes need to be made. I wanted to also say that this is actually the second attempt with this cohort to join to electronic healthcare records, and I wanted to kind of talk about the timelines involved in that as well, to share the experience. So we previously wanted to join the COVID symptom study biobank data with healthcare records via the Open Safety platform, which is another platform started during the COVID pandemic. And we had a very long contract agreement process, which hopefully we've learned from for this process we're going through now, which will hopefully speed things up. But that took around 12 months. So even after this process, unfortunately, the Open Safely had to withdraw because of budget constraints. They weren't able to support the work anymore. But that was kind of it. That process itself was almost 18 months. So reflections on where we're at so far. Yeah, these kind of linking processes necessarily contain many different steps, and in our experience, we've had long times between them, and it's kind of been a step by step. But we're hoping from here on in, we're going to run things more in parallel to try and speed things up, hopefully. But we found that the LLC platform has been really rigorous, and the communication with the team's been great. And I think when we're on, it's going to be a really good resource to use. But I guess we've wondered whether resource constraints play a role, especially in terms of how long the process has taken, given that all of this work is kind of done up front before we are invoiced at the end, and wonder if there's learning to be done from that. And it's kind of a question in our minds. We don't know the answer, but we wonder how to make this process less burdensome if possible, and faster, so we can do our research. And so that was kind of a behind the scenes from us, but we also want to know if there's any other aspects of this process that would be useful for us to share in future webinars or events about this process, so others who are looking to do similar things can learn from it in the future. And that's me. Thank you.
Thank you very much indeed, Nathan. So you're talking, it was making me think of the app iceberg, but this is the linkage iceberg, and it's perhaps even bigger, isn't it? It's a challenge, and we're very grateful to you for going through some of the pain in doing the linkage and sharing the lessons learned so that other people might do it more smoothly in the future. So thanks very much. We're nearly at time. I'm just going to share a couple of last thoughts. The Connect event, our first annual face-to-face meeting, is on the 7th of February at Google in London. Tickets are very, very nearly sold out, and I think we'll be locking the ticket sales in the next week or two before Christmas. So if you are interested in attending, come along. You can find out more details on the website. I have got the agenda there, but we don't have time to go through it. It's available on the website. You have the opportunity to sign up for advice clinics. So we have information governance clinics that are running now. You can sign up via the website, and that's also true for technology clinics and for guidance about patient and public involvement and engagement. So please do make use of that, and we hope that we can help enhance the quality of the work that we all do as a community. Final slide is to just direct you to a range of different resources here, including the website. There will be a survey after the end of the webinar. I'd be really grateful if you would fill that in. We're planning a grant writing retreat in the summertime. That's to help people put together good applications for hypothesis-led research using smartphones and wearables. If you have thoughts about what you would like within that grant writing retreat, please do fill in the feedback form so that we can design it for your benefit. And that is it for us for this December webinar. Thank you very much indeed for coming along. Many thanks to the speakers for all of the questions, and we'll see you next time in January when we've got a presentation about physical activity from Soren, Braj and colleagues. Thank you very much indeed.